What is bioprinting? 3D bioprinting is a technology where bioinks and biomaterials mixed with living cells are printed in 3D to construct natural tissue like three-dimensional structures. Bioprinting occurs in three stages. Number one, pre-bioprinting. So this involves the production of the digital model that will later be printed and the selection of the materials that will be used. Images are usually created with computerized tomography or magnetic resonance imaging or MRI, and once finalized, certain cells are isolated, multiplied, and combined with the selected bioink. Second is bioprinting. So the cell-loaded bioink is placed into a cartridge and the necessary print heads for creating the target structure are selected. Post-bioprinting. Most 3D bioprinted structures need to be cross-linked to become fully stable. So cross-linking is usually done by treating the construct with either ionic solution or UV light. Typically, you will now proceed with covering the 3D tissue models with the relevant cell medium and place it in an incubator for cultivation. This was all. Thanks for watching.